We're going to continue with AP Biology Unit 6, Gene Expression and Regulation. And in this video, video 4, we're looking at regulation of gene expression, as well as gene expression and cell specialization, which is 6.5 and 6.6. So let's get started talking about regulation of gene expression. So I want you to think about what is the advantage of regulating gene expression. So a reminder that gene expression is going from DNA information into production of a protein. That's gene expression. So what would be an advantage of being able to regulate that expression? And think about both unicellular as well as multicellular organisms and advantages for regulation. Being able to regulate gene expression enables an organism to control when and where a protein is produced. This is really helpful for both multicellular organisms that have to control in which cells different genes are active, as well as in both types, both multicellular and unicellular, is needing to regulate when genes are active based on current conditions. And what this allows is avoiding wasting energy, so not needing to produce proteins that aren't needed. It also avoids creating products that are actually detrimental, so creating protein products that are harmful in either that time or that place in the organism. So we're going to go through prokaryotic regulation first, and then we'll look at eukaryotic regulation. Just like we've seen before in terms of expression, prokaryotes are a bit simpler. Let's look at this diagram and figure out what are two methods an organism could regulate the production of tryptophan. In other words, here this diagram is showing how the general steps of making tryptophan. How could we regulate that process? That process could be regulated by either regulating gene expression or by regulating one of the enzymes itself. We've talked about the enzyme regulation in earlier units, and so in this unit we're going to be focusing on the regulation of gene expression. Why does it make sense that both of these regulation methods are types of negative feedback? And you can see that from the diagram. The idea here is if the organism already has tryptophan, it's not going to want to make more tryptophan. So it's an advantage if an organism is able to regulate this process by either decreasing the amount of enzyme activity or decreasing the amount of gene expression that codes for one of these enzymes. Both of these are going to help avoid wasting time and energy making tryptophan that isn't needed. So how do organisms do this? Specifically, how do prokaryotic organisms regulate their gene expression? And the answer is operons. So an operon is a unit of genetic function found in bacteria as well as viruses, and it consists of a promoter, an operator, and a coordinatedly regulated cluster of genes whose products function in a common pathway. As you can see, that's a mouthful. So let's translate that. Let's put that in simpler language. What does this mean? An operon is a prokaryotic control system for the expression of a group of genes. So you've got a group of genes and you want to control when they're on and when they're off. And what I mean by on and off is whether or not they're producing a protein. So in the diagram up here, which genes are being controlled in the operon shown above? This particular operon that we're seeing controls the expression of genes X, Y, and Z. X, Y, and Z are known as the structural genes. These are the ones producing the protein of interest. That's what we want to control the production of. So by controlling the activity of those genes, by controlling whether or not they are going to produce mRNA, whether or not that mRNA is turned into protein, this controls how much protein we get. So an operon enables an organism to control how much transcription takes place. Take a look at this diagram again and specifically look at the promoter. What type of biomolecule is the promoter, and what do you think the promoter's function is? And maybe use the name of it to help guide you there. A promoter is a region of DNA. You can see it's on that sort of tube-looking thing that's representing DNA. So a promoter is a nucleic acid, and that's where RNA polymerase binds to begin transcription. So it promotes the beginning of transcription. In this diagram, if RNA polymerase binds to P, the promoter region, genes XYZ will be transcribed. And so in operons and prokaryotes, this is sort of a single switch that turns on a whole bunch of lights. So rather than have them each controlled individually, they're controlled as a group. The operator is a region of DNA between the promoter and the structural genes. What do you think its function is? This is a little trickier. An operator is a region of DNA in an operon where a repressor can bind. So you notice that because it's between that promoter region and the structural genes, it can serve as sort of a block. An active repressor blocks RNA polymerase so the structural genes cannot be transcribed. What type of biomolecule do you think a repressor is, and what do you think creates the repressor? A repressor is a protein, and it's a protein that inhibits gene transcription by blocking the RNA polymerase. In this diagram, the repressor is produced by the regulatory gene. You can see that in yellow labeled I over on the left. So in this diagram, the repressor is produced by the regulatory gene. If that is active, the repressor can bind to the operator region of the DNA and block RNA polymerase. So repressors sometimes have active and inactive forms. If the repressor is active, it is blocking the transcription. It is an active repressor. We're going to look at two examples of operons that function in slightly different ways. And these examples are lac and trip. So in a lac operon, structural genes produce enzymes that break down lactose. So in that generalized diagram, structural genes XYZ, think of those genes as producing enzymes that break down lactose. In this case, when should those genes be active? Sort of from a fitness standpoint, when would you want those genes to be turned on? And when should the repressor be active? And then I have the same questions for thinking about the example of a trip operon. So in this case, in those structural genes, in this example, those structural genes are going to produce enzymes that create tryptophan. In this case, when should the genes be active and when should the repressor be active? In the case of the lac operon, because the structural genes produce enzymes that break down lactose, you want those to be active if there's lactose. You want them to be inactive if there's no lactose, which means you want your repressor to be active if there's no lactose. So basically, gene expression should be on if there is lactose because the structural genes make an enzyme to break down lactose. So that's when you want it working, is if there's lactose. The trip operon is just the opposite. In this case, the structural genes are producing tryptophan, or I should say they're producing enzymes that produce tryptophan. But the end result is when those genes are active, you get more tryptophan. So you want those genes to be turned on and transcribing in the case if there's no tryptophan, right? You need more tryptophan if there's no tryptophan present. Likewise, you want your repressor to be active if there is tryptophan present. And you can see from the diagrams that there's going to be a slight structural difference in how these are operated. So in the case of the lac, we have the inactive repressor when it is bound to a molecule. And in the case of the trip operon, our active repressor is only active if it's bound to the molecule. So let's go over those again. A lac operon is also known as an inducible operon, which means that in most cases, it's off, right? The gene expression is off. And so the sort of baseline repressor is active, which means the repressor is going to be blocking the transcription. However, if we need it to be turned on, it can be turned on. And we need the lac operon to be turned on if we have lactose. So what lactose does, and you can see that's represented by that green ball there down at the bottom, the green circle, if a form of lactose is present, that binds to the repressor, so then the repressor no longer blocks the transcription. It's kind of like we've taken the brakes off, and now the RNA polymerase can access those genes, and the transcription process happens. So again, if it's needed, it can be turned on, and it's needed if lactose is present. The trip operon is the opposite. This is a repressible operon, which means the default position is that the genes are on. They're being transcribed. So in this case, the default position is we make the enzymes to make more tryptophan. But if you have tryptophan, you want to be able to turn this off. So that means that tryptophan will activate the repressor. So if there's tryptophan in the system, you will stop the production of the enzymes that make more tryptophan. In both of these cases, in both the lac and the tryp, it's to save resources. You don't want to be making a product you don't need. So as a review, here's a table showing both of these cases, both of these operons, and how their repressors are slightly different in terms of when they're active. But both of these share the overall process is that they're both considered forms of negative control of gene expression. And that's because they're related to the braking system, the repressor. What do you think might be a positive control of gene expression? A way of positively controlling gene expression is stepping on the gas. And the gas region is essentially the promoter. So when you need really a whole lot of something, you want to step on the gas and make more of it. So let's look at both of these diagrams and see if that makes sense. Explain what's happening in both diagrams. In both cases, lactose is present, so that means the lac operon genes are active. So we can think about that as our repressor system. Our braking system is off. So the brakes are off, right? That means the genes are active. But on the left, glucose is in short supply. That means the organism has additive incentive to break down the lactose so that the operon increases its activity by activating the promoter. Let's think through this. Lactose and glucose are both types of sugars, but glucose is a simpler sugar. Lactose is more complex, right? It has a larger structure. So you only want to take the time to make the enzyme to break down lactose if you really need it. If you have glucose around, you don't really need to break down that lactose quite as much. But if there's no glucose present, you better step on the gas and really get to work breaking down that lactose. So as a review, negative control of the operon relates to the repressor. You can think about that like the brakes. Positive control of the operon relates to the promoter. You can think about that like the gas. Now let's go on to eukaryotic regulation. Eukaryotes, as I was saying, are much more complicated. We were thinking about an operon as kind of like turning on one switch and a whole bank of lights goes on. You turn it off, they all go off. Eukaryotes, they're individual switches for individual lights or genes, and in a lot of cases, you can really fine-tune those. So you can almost think about them as being like on individual dimmer switches, whereas operons are generally sort of an on or off kind of situation. So in eukaryotes, something else to keep in mind is that in multicellular organisms, almost all of those cells are genetically identical. But the cells are different. So that means the difference between the cells is due to differential gene expression. We can regulate this gene expression in many different stages, and that's what this diagram on the left is showing. Something else good to be aware of is that abnormalities in gene expression can lead to diseases including cancer. So understanding this process is really important to us. It's really important to understanding how things are working sort of on a daily basis, and then what happens if they're slightly off. We're going to look at each of these 6 types of gene expression regulation. So if you look over on that diagram on the left, you'll see 1 through 6. We're going to go through these one at a time. So let's start in the nucleus where the DNA is, and this is chromatid remodeling. So here you see, and this is a review from earlier in terms of chromosomal structure, the DNA is wrapped around proteins called histones. Here we have 2 processes that can affect that wrapping process. So we have methylation and acetylation, and those can both be forms of epigenetic inheritance. What does that mean? Epigenetic inheritance is the inheritance of traits transmitted by mechanisms that are not directly involving the nucleotide sequence of the genome. So traits can be still transmitted from parent to offspring, but not in terms of the nucleotide sequence. Instead, we can get information in terms of the wrapping process. So if you see in methylation, if an area of the genes or an area of the DNA is methylated, the DNA is sort of more packed tightly together, whereas acetylation, it's opened. And that is going to change gene expression, because if it's tightly coiled, the transcription can't happen. Whereas if it's loose, then transcription can happen. So having access to different genes can change all sorts of things, including behavior. An example of this is that the type of parenting a baby mouse receives can change the methylation of some of its genes that then affect its behavior as an adult, which then can affect how it parents its own baby mice, which can then kind of continue down the generations. Methylation patterns can be inherited. Let's go on to number 2. So here we are in the process of transcription. So here we're going from instructions of DNA to produce a molecule of RNA. Let's start by naming the type of biomolecule and the function of activators and enhancers. So see if you can figure out anything from this diagram. I recognize it's complicated, but see what you can figure out about activators and enhancers. Activators are a type of regulatory transcription factors, which just means they help control when transcription is on and off. So they are a type of protein, and they bind to a specific region of the DNA called the enhancer region. When the activators bind to the enhancers, they allow for transcription to happen, and that allows RNA polymerase to begin transcription. So you can see in this diagram that the DNA is not being transcribed until all of those activators bind to the enhancers. And once they do, if you get all those proper activators in place, then transcription begins. The whole complex comes together. A couple things to point out on this diagram. The TATA box, or TATA box, is a type of promoter sequence which specifies to other molecules, such as RNA polymerase, where transcription begins. The TATA box is named for its conserved DNA sequence, which is most commonly TATAAA. I think I said too many A's there. You don't need to memorize that. But just recognize if you ever see that term, that's a specific region of the DNA where the RNA polymerase latches on. It's showing the RNA polymerase where to latch on. All right, so to continue our understanding of transcription regulation, because this is really an important part of overall gene expression regulation in eukaryotes, let's take a look at this diagram. What's going on here? So what this is trying to show is that there's a liver cell shown on the left and a lens cell shown on the right. In both of these cases, we've got the same DNA. But not all of those genes are going to be active in both of the cells. In the liver cell, you need some genes to be active, and in the lens cell, you need different cells to be active. So even though liver cells and lens cells have the same DNA in their nuclei, different genes are going to be transcribed. And the reason, or sort of the mechanism of controlling this, is that there are different available activators. So you can see those little molecules sort of floating around. Those are proteins, and the available activators are different. So the activators in the liver cell will cause the albumin gene to be expressed, and they're not the proper activators to activate the crystalline gene. Whereas in the lens, you get the opposite. The activators do not bind with the enhancer regions of the albumin gene, so that gene is not turned on and transcribed and expressed, whereas the crystalline gene is, because those activators are present in the region. So which activators are present, what proteins are available, controls the beginning of transcription, which eventually then controls what proteins we have. Just a note on this diagram, because I know this diagram can be kind of confusing. To save space, the diagram doesn't show the folded DNA to the left of the activated genes. So remember, there was that transcription sort of complex that involved the DNA folding over, like we saw in the picture before. This diagram is just trying to save space to not show that whole region. All right, let's go on to the next step of eukaryotic gene regulation. So after transcription has happened, then we get an RNA molecule. Remember, in eukaryotes, one of the steps of transcription is RNA processing. Let's do a review. What are the 3 modifications that take place during RNA processing in eukaryotes? Those 3 processes are the addition of a cap, the addition of a tail, and splicing together of exons. So we cut out the introns and we keep the exons. And as a reminder, that process can happen in multiple ways. So alternative splicing allows for the increased diversity of proteins. The next step in, or the next possible mechanism of regulating eukaryotic gene expression is RNA degradation. So degradation just means falling apart, right? Why would an organism want the RNA to degrade? Organisms don't want to continue making a protein product forever. So again, we need to have a way of controlling sort of the on and off switches in lots of different subtle ways. So breaking down the RNA is a way to decrease the amount of protein product that comes from that mRNA. Do not worry about the details of this diagram. It's just a visual to help you understand that RNA can be broken down. Don't worry about all these details. Another way that eukaryotes can regulate their gene expression is through translation regulation. Again, here, don't worry about the details. Just recognize that translation can be regulated. You may encounter some of these terms of small regulatory RNA, such as microRNA or small interfering RNA, but I wouldn't focus on those terms. Just recognize this is just another way in which eukaryotic gene expression can be regulated, is by regulating whether translation ever happens at all. The final step of eukaryotic gene regulation is post-translational modification. So after we get our protein, we can still control the process. How does that happen? What's this diagram showing? This diagram is showing that phosphorylation, which is the addition of a phosphate group, can cause change to the shape of a protein. And since form influences function, a change in shape can cause activation, deactivation, or change in behavior of proteins. So in terms of the overall control of which proteins are active, you can use phosphorylation. In this case, you can see on the left a non-phosphorylated protein, and on the right a phosphorylated protein. Because that was a lot, I just wanted to return kind of where we started from. Think about all of these different mechanisms have one overall goal, which is to improve fitness by controlling which genes are expressed when and where. We can see the importance of gene regulation when we start talking about embryonic development. So here we're moving on to Unit 6.6. Embryonic development involves three main processes: cell division, cell differentiation, and morphogenesis. What do you think is happening in each of these processes? Cell division is making more cells, so this is mitosis. Cell differentiation is cells becoming different, becoming specialized in both structure and function. Morphogenesis is giving an organism its shape. We're going to focus on cell differentiation in this unit. Keep in mind that all of the cells have the same genes, so in order to get different cells, we need to have different genes expressed. For example, we can have the presence of different activator proteins in different cells, as we saw in the regulation of transcription in eukaryotic organisms. But how do these differences start? How do cells end up with different activator proteins in them to begin with? To answer that question, let's look at cytoplasmic determinants. Based on this diagram, try to define that term. Cytoplasmic determinants are maternal substances in the egg that influence the course of early development. Keep in mind that the egg is quite a bit larger than the sperm, and so carries with it a lot of additional material rather than just the genetic material. So cytoplasmic determinants include mRNA and proteins such as activators and other transcription factors, and all of these things can influence early gene expression. If you notice in this diagram, that unfertilized egg has different cytoplasmic determinants in it that are unevenly distributed throughout the egg. So on the left, you see more of those sort of orangish circles, and on the right, you see more of those green triangles, which means that in those early cells, there are going to be different proteins, or whatever the cytoplasmic determinant, but there's going to be different ratios of those cytoplasmic determinants in those early cells. Cytoplasmic determinants lead to differences in cell function in the early embryo. What happens then? Take a look at this diagram. What is induction? Induction is a process in which one group of embryonic cells influences the development of other, usually causing changes in gene expression, which means that early differences can set up sort of cascading effects in some of those later cells of the embryo. You can see here that there's a signal molecule coming from one cell and received by the other cell. That might trigger a signal transduction pathway that ends up in differential gene expression in the nucleus of that next cell. So behavior of a neighbor cell can influence a cell. Let's see if we can make sense of this. This is complicated stuff. I think an example is going to help. We're going to start looking at something called bicoid. So bicoid is an mRNA molecule located on one side of the unfertilized egg in Drosophila. So you can see that in the diagram on the left. How might the localization of bicoid mRNA influence the distribution of bicoid protein in the embryo? Where there's a lot of bicoid mRNA, you're going to get a lot of bicoid protein. So this creates a gradient of the bicoid protein in the early embryo. There's going to be more bicoid protein on the side where there was more mRNA. How might the concentration of bicoid protein influence the development of the cells in these different regions? So in that picture on sort of the center on the bottom, you can see that this gradient can be sort of thought about in chunks. So how can this gradient influence the development in these different regions? The gradient of the bicoid protein can lead to the determination of body plan. So if there is a certain amount of bicoid protein, that'll turn on the expression of one type of cell. If there's a different concentration, that'll turn on the expression of a different type of cell. So if you can see here, this is in normal development. We have bicoid mRNA, and where that bicoid mRNA is where you're going to have the highest concentration of bicoid protein, and that's going to trigger cells to express different genes that are sort of normally expressed in different regions of the Drosophila. Don't worry about the terms on the very ends. Those are sort of the very end of the head and the very end of the abdomen. But understand that on the left side here we have the head, and on the right side we have the tail. So the amount of bicoid protein present determined what body region type of cells would be activated in that region. In hopes that you understand this, or to sort of check if you're understanding this, let's make a prediction. What do you think would happen if scientists created an embryo that didn't have bicoid at all? If there's no bicoid, you're not going to get a head. Because remember, it was those high concentrations of bicoid that trigger the development of a head. So if you have a bicoid deficient mutant, you're going to end up with two tails. Let's do some more predictions. Take a look at the diagram here in these three types of experiments. What's going to be the result of each of them? On the left, you have a mutant that doesn't produce bicoid, but the scientist adds bicoid. That's going to end up normal, right? It's going to develop normally. So you're adding back in what you already sort of took out. So you take something out genetically and then add it back in physically, and it restores the sort of normal phenotype. What if you add it back in, but you add it back into the wrong spot? In this case, you're going to end up with a head right in the middle, because remember, the highest concentrations of that bicoid protein are what trigger the expression of the genes that are found in the head region. And what if you have a wild-type organism that's sort of expressing bicoid, quote-unquote, normally, but you add some extra bicoid to the other end, you're going to get two heads. And so in this case, you're going to have highest concentrations of the bicoid on both ends and the lowest concentrations in the middle. So I know that might have seemed complicated, but it's actually a fairly simple example of there's a whole host of genes that operate kind of in this way. So there are body plan genes, and then there are cascades under these body plan genes. So they're sort of master control genes for a region that then influence genes underneath that. An example of genes that help control body region are Hox genes. So Hox genes are active in different segments of the body. Here we can see that there are Hox genes in one area of the body that turn on or activate antenna genes. And then there are Hox genes that are active and produce proteins in other areas of the body. And those are sort of activating the leg genes. What do you think might happen if a Hox protein that is usually produced in the thorax region is added to the head of a Drosophila? And then you end up with flies growing feet out of their head. So antennapedia means antenna foot. Antennapedia mutants are flies that have legs growing where most flies have antenna, and you can see that on the right. So scientists learn a lot about gene function by seeing how gene products influence development by sort of doing these manipulations to see what happens if we take this gene product and we put it over here instead, what's going to happen. So I'm going to leave that off with the fly growing feet out of its head, and we'll continue to learn more about gene expression in our next unit.